Greetings, life science grade core of learners. We are meeting this time of the year, uh, pushing our last session, taking the last push of the year. I want to appreciate your connection and your utilization of time and data connecting to Metric Hub. I promise you that your exams are not gonna be the same. You are going to go to exam smiling and happy. When you write this paper, just go ready. Don't be nervous. Just be comforted. Feel comfortable. This is the last session, like I said. So please, most importantly, when you are writing an exam, always know that you read instructions first. Uh, this paper that we are going to do today is just a complete, interesting piece of paper that you are going to see all the necessary or all the relevant areas of concern which actually appears or which makes part of your syllabus that I'm quite sure that is going to be very useful to you. Just be patient and follow the presentation to the end. You are seeing that this is a comprehensive presentation. It's a comprehensive coverage of the entire paper or of the entire syllabus of paper one life sciences. So what we are saying here is that this is just a replica of our national paper that we're gonna write. Let's follow and relate to all the questions that are coming here in. Without wasting any time, I want us to go straight into the paper and then just see what is it that we are supposed to answer and be able to understand and expand the relevant areas of concern that would arise. So which structure protects the entrance, which structure protects the entrance to the vagina? We are saying when you look on female reproductive system, I'm sure that you know when you are going to exam, there is no way which you are going to go to exam and then you cannot understand human reproduction. It actually constitutes the best and the most marks when it comes there. So human reproduction, the male and female reproductive structures, you must be able to know them. So which structure protects the entrance to the vagina? Entrance, if you look at the entrance, we are saying the cervix, it is not protecting the entrance. Remember, it is inside which is responsible for labor pains during pregnancy. The cervix, it is there for contraction and relaxation of the vagina and reproductive organ to prepare for to prepare for birth. Anus, it is not related or connected to the vagina. The urethra, it is a male it is on the penis. So what we are saying is the structure that protects entrance to the vagina is the valve, which we say again is called the hymen. In other words, the valve which is also used to determine virginity when we talk about women or when we talk about female reproduction. The valve is a seal that is found at the entrance of the vagina to signify also that the woman is not yet known a man, and then it protects the entrance to the vagina. That is B. And then in humans, fertilization usually takes place in the what? Where do we see fertilization occurring? I'm quite sure that if you refer to the channel again, this we have discussed and explained that it is in the fallopian tubes where we see reproduction, I mean, where we see fertilization occurring. The fallopian tube, or they can also say the oviduct for clarity's sake, the oviduct or fallopian tube, that is where fertilization occurs. Uterus is for implantation. Vagina, it is the birth canal or also the excretion canal or also the copulation organ. And then ovary is where the female reproductive hormones and eggs are produced. So the one where fertilization occurs is the oviduct. Number three, which part of the brain is responsible for voluntary movements? Voluntary movements, we said that you need to understand that the human brain, It is also on the channel. Please go and check Metric Hub, the human brain. It is well explained and also given the functions. So the part that is responsible for voluntary movements, we are saying voluntary means plant. We are saying it means plant is to say intentional, or it means conscious. We can also say plenty, or we can also voluntary. We can say conscious, as to say the person is involved. You decide, the person decide what to do. So voluntary movements are controlled by the cerebrum. We say the cerebrum controls voluntary movements and also the cerebellum. So A or B can do that. So I'm sure that you remember that when you go and look on human brain, we say that the biggest part of the human brain is the cerebellum, and then it the the cerebrum is the biggest part. The cerebellum, it is found on the human stem as a brain stem, which is next to the medulla oblongata, connecting towards the spinal cord. The hypothalamus, it is something that is, it's a it's an endocrine uh, gland also, which we are saying that it's not responsible for voluntary movements. So please go and research and read on human brain from Metric Hub. It is quite explicitly explained which disease occurs when blood sugar levels are too high. I'm sure I remember the issue of human response to the environment. We talk about various diseases and also uh, the way they are treated and so forth. So when you look at disease, blood sugar levels, we talk about the balancing of sugar in the body, which is balanced because of insulin glucagon dynamics, this. Which controls the glucose levels. So what I want you to understand is that when you talk about hyper, it means something is what it's in excess, and then excess of any given condition or a hormone or a substance, we say that it is hyper, and then hypo, it means very low, extremely low, which is the opposite. So number four, the disease which occurs when blood sugar levels are too high, it is the blood sugar levels, it causes diabetes. I'm sure if you remember the types of diabetes, we are saying diabetes 1 and diabetes 2, which we say that they are caused because of excess sugar levels in the body, which is because of inadequate or insufficient insulin secretion, which must convert the glucose into into glycogen. So what we are saying is that these conditions, you need to go again on Metric Hub, consider that, and go and research, read very well. Iron, rickets, it's caused by a lack of iron and calcium in the body, nutrition deficiency of iron and calcium, and then of course goiter, it is the enlargement of the human throat, which also happens not because of blood sugar levels. But then what I want you to understand is hyperthyroidism is excess thyroxine hormone. It is a condition which causes excess secretion of thyroxine in the body. So it causes some other related conditions, which we can also want us to say that go and consider reconnecting to Metric Hub, the structure that joins the two hemispheres of the brain. It is called the corpus callosum. The two hemispheres of the brain are balanced or connected by the corpus callosum. The corpus callosum, we find it there inside the brain, connecting the two hemispheres. I'm sure that you remember that the right and left hemisphere, which we say that they are connected by corpus callosum, and then the pituitary gland, which we said is the master gland, which secretes a mass of hormones, which are also responsible for various conditions in the body. Medulla oblongata, it is part of the brain that is an extension of the spinal cord, which is found connecting to the head there. So pupillary reflex is the ability of a typical human eye to adjust to different light intensities. That is it in the environment. So we said that there are stimuluses that causes various sensations. Human senses are caused and controlled by various stimulus. Each of them has got its own stimulus. So uh, sight is controlled, or the stimulus for sight is light. Light enables us to see. Which combination of processes will occur in a typical human eye when the light intensity changes from dark to bright? It has been dark and then it is going bright. So now what we need to understand is the pupillary reflex mechanism that has to take place in terms of this condition. What happens? So what we need to understand is that the radial muscles contract. We are saying from bright to light, from dark to bright. So what we need to understand is that one, the circular muscles must contract. The circular muscles must contract, which is two. And then we also need to understand that the pupil constricts. The pupil constricts in order to reduce the amount of light, to reduce amount of light entering in the eye, to reduce light intensity. Light intensity is reduced because of the constriction. Constricting means becoming small. And then we are saying again, four, radial muscles relax. The radial muscles relaxes, which is also correct. So what we are saying is that number 6, our correct answer is A, which we said that is circular muscles contract, the pupil constrict, and the radial muscles relaxes. I am saying go and look again on Metric Hub, the functioning of human eye, accommodation, pupillary mechanism, and binocular vision. These are the 3 ways through which a human eye works: pupillary mechanism, accommodation, and the binocular vision. How do these promote effective sight and also functioning of the eye? So number 7, which of the following can be used to treat a severe mid to ear infection? We are saying ear infection, that is used to treat severe mid to ear infection. We talk about diseases of the ear, or conditions that can cause ear not to function properly. So now, when the middle ear is infected, what must be understood is that we use a grommet. A grommet is used. A grommet is used, which I said again that if you look there, it is used in order to ensure that it serves to ensure that the middle ear infection is still working, and also enable the creation, or the stimulation of the eardrum, in order for it to cause the development or the stimulation of impulses that must be transferred to allow hearing. So it is a grommet that must be used to treat middle ear infection. A person experiences the following symptoms: loses weight easily, or loses weight easily, is always hungry, never feels cold. So in other words, never feels cold, we can say always have fever, or we can say that the temperature is constantly or usually high. So the most likely explanation for this combination of symptoms is that what is causing the person to lose weight easily, always hungry, has got high appetite, and then we are saying there is always never feels cold. So this is caused because of excessive thyroxine hormone. So he has an overactive thyroid gland. Overthyroid gland will stimulate, or will cause metabolism, also will cause also high metabolism. Digestion will be very high or will be very fast because we are saying thyroxine hormone that is secreted there will always cause also the person to be always hungry because of extensive rate of digestion, and then it would also cause the person to lose weight because again there is extensive maybe quick flushing of the food without enough absorption of the nutrients. Secretions are released into a cavity or onto the surface through a duct in the body by uh, we are talking about types of this can be related to the types of hormones, the types of glands, the exocrine and the endocrine. We talked about the ones that hormones are carried through ducts, and also where hormones are directly secreted into the blood, which are carried duct we are saying there is no ducts, there are no tubes. So secretions are released into a cavity or onto the surface through a duct in the blood by it is because of the exocrine gland. The exocrine gland, it is the one that actually allows or that releases secretions. We are talking about hormones. Hormones, we say that they are secreted because they need to be released in microscopic amounts, very limited amounts because they are very powerful. So let's understand that when they are carried through it entails exocrine glands. And then I've also given this on the channel on explanations of, on differences between endocrine and exocrine. Endocrine, it means they are released into the blood straight. There is no ducts that are used to carry the hormones to their target organs if they are endocrine. So the nervous system is just that rapid autonomous response of the body towards a stimulus through nerve impulses. And then which of the following occurs when the sympathetic nervous system is more active than the parasympathetic nervous system? I also have an interesting presentation on channel on the differences between sympathetic and parasympathetic nervous system. So now, which of the following when the sympathetic nervous system is more active? So we are talking about when the sympathetic nervous system is is very active. So it is actually it causes increased what? Increased breathing rate. We say that the sympathetic and parasympathetic nervous system, what happens is that the two systems, they work opposite to each other. We say that you need to understand that when this occurs, uh, increased breathing rate takes place. So we talk about the dilatation of the eye. We talk about all these instances whereby there is an increase of activity in sympathetic nervous system. Parasympathetic system, we say that it helps to control the rest and digestive processes. To say that it works effectively when a person is inactive, and then it is actually there to increase the liberation of energy from food eaten. So now what we are saying is that increase in digestion, it takes place during parasympathetic nervous system. We give the biological terms for each of the following descriptions that are given. 1.2.1 sharp structures found in the plants for protection from herbivores. We talk about plant protection mechanisms. We talk about the chemical, we talk about the physical, and also other ones which we can say biological, which is otherwise through the secretions or through the smells. So now what we are saying is that sharp structures found in plants for protection from herbivores. I have also a channel, a good presentation which talks about the thorns which are there or which are found on some types of vegetation where we can tend to have leaves growing inside the thorns. The leaves are very tiny and they cannot be eaten by animals because they are inside the what? They are inside the thorns. So this is called, we say these are the thorns. We call thorns. Those are, they are there to actually prickle. They are there, thorns, they actually cause prickle herbivores, so then they cannot eat them. So we call that, these are called what? Thorns. Thorns, they protect, the leaves are very tiny and they are hidden in between the thorns there, such that animals cannot eat them. Plant defense mechanisms. We talk about plant defense mechanisms. That is under plant response to the environment. Go and check on it again. Metric Hub is enough for you. Type of vision that results from having two eyes. It is called binocular vision. I was talking about the functioning of the eye. The type of vision that results from having uh, two eyes, we call that binocular vision. The ability to see with both eyes, which we say that it gives 180 vision that has to be found there. Binocular vision, a hormone produced by pituitary gland that stimulates milk production in humans. Milk production is actually stimulated or promoted to come as a result of prolactin hormone. Prolactin, it is because of prolactin hormone. It stimulates the mammary glands of animals to produce more milk, which actually is it is secreted from the time of pregnancy, continuously going with regard to the development of pregnancy. And then a condition where the cornea is uneven, resulting in blurry vision. This question class, it is quite very common. Number 4, we see that it has been surfacing quite a lot. It has been coming again and again, which obviously needs to be understood, that we need to understand the astigmatism, which we say that the cornea of the eye, the unevenness of the cornea of the eye, which results in blurry vision, it is called astigmatism. Astigmatism is very important for you to take note of that. Astigmatism. And then the response of system of stems towards a light stimulus. We talk about types of tropisms. Types of tropisms, we said we have got photo and geo. So this is called, with regards to light, it is called phototropism. Phototropism, which is the response of plants stems towards light. And I have given this again on the channel where we talked about types of tropisms, which I say that we have got negative phototropic and negative geotropic, which we say that the negative is when we talk about when we are under photo, we say the plant roots are negatively phototropic. They move away from what? Negative, meaning to say that the part of the plant, it moves away from a certain stimulus. So if we say negative phototropic, we need to know that it is the roots which develop or which grows which grows downwards away from light. We say that they are negative phototropic, and then the stems also are negative geotropic because they move away from the what? From the force of gravity. So I want you to know that types of tropism, we have got photo and geo. Geo is towards gravity, which is of the roots. A hollow ball of cells formed from a hollow ball of cells that is formed from the zygote. We said a zygote is just a fertilized egg. It's a fertilized egg. That is what we call a zygote. And then we say when it is a hollow ball of cells, we call that a blastocyst. It's called a A blastocyst. I remember also that there are questions which comes to say identify or put it as a flow diagram that identifies the stages of from fertilization to birth, to say from fertilization to birth, which you need to know that what are the developmental sequences or developmental stages that you need to take note of there. So that is it. I'm sure that you are following, and it's quite interesting. Let's do it together and let's get the best marks. With question 1.3, I'm sure you know the instructions there. It is either applying on A, B, none, or both. So 1.3.1, the hormone that is in excess in an abnormally tall person, a person who is abnormally tall, we call that, we talk about aglomegaly. Aglomegaly, which is caused by, it is caused by growth hormone. We are saying it is caused by growth hormone. Growth hormone, it causes the extensive elongation of bones and muscles, which we say that it causes a person to be abnormally tall, which is what, which is B. And then luteinizing hormone, I'm sure that you remember this one is for what? Is for ovulation. It stimulates ovulation, uh, luteinizing hormone. And then corrective lenses for long-sightedness. Long-sightedness, it is corrected by convex lenses. I have got also, I have seen in the question paper here that we are going to talk about ways of correcting at visual defects. So long-sightedness, we talk about a person who has got to use, he has to use convex, he has to use convex lenses. The convex lenses are used in as a remedy towards long-sightedness, and the opposite is short-sightedness, which a person can only see things that are near, which can be corrected by the concave lenses. So sometimes we would say that the image falls within the retina, and then on long-sightedness, the image falls behind the retina. So we need to use convex lenses in order to increase the refraction of light such that the image can fall within the retina and on the spot. A disease of the central nervous system. A disease of the central nervous system. It is actually we say multiple sclerosis and Alzheimer's disease is actually both of them. This is both A and B. Both A and B. I'm sure I remember the multiple sclerosis. We talk about the hardening and cracking of 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 uh of the neurons, which also causes the neurons to be uncovered, which we can find out that there is a retardation in the speed through which impulses travels through a neuron. So Alzheimer's disease is also another disease which is caused because it's a central nervous system disease which also causes degeneration of brain cells. So study the diagram. It's an amniotic egg, and answer the question that follow. I've said that this is also quite an interesting one. The development of an amniotic egg. We talk about reproduction in invertebrates, which we say that it facilitated evolution. It enabled the habitation of places which used to be impossible, where water was not there, but now because of a shelled egg, we say that an amniotic egg is it's a shelled egg, which we can give an example of a bird, a chicken, or any other reptiles which have got eggs that are shelled. So now the question is saying, give the letter and name of the part that protect the embryo, the developing embryo from predators. The developing embryo being protected from predators what is it that protects them from predators? We say that the egg is actually covered by the shell. So it is actually protected by what? By the shell, which we say that it is shell. And C protects the egg from drying out and also predators. C, which is the what? The eggshell. The eggshell, it is the one that protects that. And then provides nutrients for the embryo during its development. Provides nutrients. Nutrients are provided are coming from or are provisioned by the egg yolk. So egg yolk provides the what? The egg yolk provides egg yolk. It is coming. It is there to what? To provide nutrition to feed the developing young one. Provides nutrients to the embryo during development. So it is F, which we say is what? Is the yolk. And then allows for storage of waste products from what? From the embryo. Waste products from the embryo are collected in the aliontos. So aliontos is the one that is responsible for that. So aliontos, you should look for it to say which one is it. Aliontos, which is given as what? Aliontos, which is given as E. E is the one that is responsible for, is the one the storage of waste. It is E, we say the aliontos, it is responsible for storage of waste products from the embryo. That is very important to note there. And then obviously we have got to know that the developing embryo is A there. We have got the amnion, which is A, which is covering and protecting the developing embryo and giving a membranic cell that actually gives or houses the amniotic fluid that should also avoid the drying out of the chick inside the egg. Different stages of development we are given. This is reproduction in what? In invertebrates. So now what is given here is that we need to understand the artificial precocial development. Artificial precocial development. I also have a beautiful presentation on the channel. Please go and verify and get more detail on that. There are quite good notes there for you. Go and check. So now the question is saying, name the diagram that represents artificial development. The diagram that represents artificial development. So what we are saying is the artificial development is whereby the young ones are born and then they are not able to look after this. Saying young ones are incapable of looking after themselves, looking after themselves from birth. So they are underdeveloped. In other words, you should know that these young ones are underdeveloped. So what happens is that there are assumptions and information that we need to understand, which is quite interesting in these philosophies or in this scientific situation that we are given here. So artificial development, it is shown by them, these young ones which are born, they are unclothed, they have no feathers, they have got closed eyes, they are dependent on parents. We can give as many as possible. And then I was saying that there are quite many other points that are given there. They are dependent on their parents for what? For thermoregulation. Yes, you can see that the mother must cover them in order for them not to die of cold. They need to be fed. They can't walk, they can't move. So we are saying it is what? Artificial development is shown by diagram what? By diagram A. Type of development where a small amount of yolk was available to the embryo. We are saying that the underdevelopment that we are attributing to artificial development, it is subject to the size of the yolk, which is also there, that the young one cannot be fully developed because the feeding mechanism which is coming from the yolk is not enough, is inadequate. So we say that A again is the one which is causing or which shows a small yolk which results in an underdeveloped young one that is hatching before it can be able to look after itself. So the precocial are born strong, they are born ready to walk, they are born able to do things. So what happens is that the yolk is enough that they can feed enough and develop strong muscles and bones that immediately from hatching they are able to come out and be able to face the world and be able to survive in their environment. They can run, they can feed, they can do all such things. I said there is quite a good example. I mean, quite a good presentation on the channel. Let's go and connect on forms of development in terms of the precocial artificial development characteristics and the assumptions thereof, which is causing such developments to be like that. We have got uterus of the pregnant woman. This is also quite an interesting one. We are given a woman who is pregnant, and then now they are. It is quite important that before we go to exam, we are able to relate and answer these questions just at the by the. I mean, the information should be on our fingertips. Human reproduction. So identify structures numbered A1, identify structures numbered 1, numbered 3, numbered 5, numbered 6. So I was saying that if you look at this, if you look at this diagram, it is also quite articulated and explained on Metric Hub. I told you that Metric Hub simplifies the journey for you. So what we are saying is structure numbered 1, as you can see, 1, we are seeing that that is amnion. Amnion, 1 is amnion, which we are saying that it surrounds the what is the amnion that is actually covering the developing baby there. It is covering the developing baby there. And then the next question is saying, we are saying A1 is amnion, and then 3, 3 which is given here. 3 is it is actually the uterus wall, or we can say the uterus. 3 is the uterus wall or the uterus. 3 is the uterus wall. You can say that is the uterus wall. As you can see there, we said that it is where the baby grows in. It is that big cavity or that sac through which a baby develops inside. And it has got also question that comes there. Explain how the uterus is adapted to perform its function. Explain how the uterus is adapted to perform its function. Please take note of that. We're saying that it has got muscles which allows contraction and relaxation to prepare for birth, and also it is vascular. We say that it has got it's a halo which is big enough to allow the movement of the developing young baby. All those, please take note of them. And then 5, 5, what is 5? If you look at 5, we can see that it is the what? It is the civics. This is the civics, which we can see here. 5 is the civics. 5 is the civics. The civics, which we said it actually, it is actually there, which actually affects or allows birth. It allows birth. It allows the labor pains. It allows labor pains. It expands and contracts to allow birth process, which is also affected because of the labor pains that we are talking about. And then we also said that there is oxytocin hormone which works on it. It is also affected and influenced by oxytocin hormone. It actually is it's a hormone that promotes labor pains, which works on the cervix. So please note that is what is the cervix. So number 6 obviously is the fetus. As we can see, that is the developing young baby, which is the fetus. There is the fetus, number 6. Just bonus marks that are given for us here. I don't think any one of you is gonna miss any marks. And then we were also gonna talk about the amniotic fluid. Give the functions of amniotic fluid. We said that it actually allows prenatal care by providing amniocentesis, the collection of the amniotic fluid, in order to examine and determine congenital disorders that can also be developing within the baby or upon the baby. So the amniotic fluid, it actually promotes those services to be offered, and also it absorbs pressure. It also avoids the drying out of the baby. It is also for physical and mechanical forces that might impact on the baby. It is actually there to also nourish the baby in terms of the flexibility to avoid disabilities. The baby can rotate and move because of the amniotic fluid. So this is quite an interesting one again. Name the structures that acts as a birth canal. Name the structure that acts as the birth canal. Birth canal, I'm sure you remember that it is through what? Through the vagina. The vagina is the birth canal. Birth canal, it is actually the vagina. I'm sure that we are quite aware that the vagina serves many purposes. An excretion pathway, it's an organ for copulation, for birth canal, and all other these factors that we are talking about, the vagina. And then secretes a hormone that maintains pregnancy. We are saying maintenance of pregnancy. Secret 1.52. Name the structure that secretes the hormone that secretes a hormone that maintains pregnancy. Maintenance of pregnancy. What is that hormone that maintains maintenance of pregnancy? It's maintained by a progesterone hormone. Progesterone hormone is there it is the one that is responsible for maintenance of pregnancy, which is secreted from the, we are saying it is where is progesterone secreted? Progesterone is secreted from, it is secreted uh, from what? From from the ovaries. When an egg is released, the corpus luteum secretes, which we said that is the remaining inner side tissue of an egg, which is after ovulation. That is where we see the secretion of what? Of progesterone. Progesterone is secreted there. And then transport oxygen and nutrients to the developing fetus. The developing fetus nutrients are transported. When oxygen and nutrients, these are transported by the umbilical vein. Umbilical vein, which we can say in other ways, somebody can say umbilical cord, but upon the umbilical cord, we have got the umbilical vein, which is for oxygen and what? And nutrients. And then umbilical artery, umbilical artery is for what? For waste and carbon dioxide. They are carried through the umbilical artery, but oxygen and nutrients, these are taken to the baby, to the fetus, and these are taken from the fetus. You need to take note of that. Umbilical artery, it carries the waste from the baby, and then the umbilical vein, it carries from the mother to the what? To the developing fetus. And then name the tissue of the uterus that contracts to enable the woman to push the baby out during birth. The tissue of the uterus. Name the tissue of the uterus that contracts to enable woman to push the baby out during birth. It is actually the muscles. It is the muscle which is responsible for the pushing of the baby out of the, to allow birth process. And then several functions are listed below. We are given several functions that are listed below. And then the question which is there is saying, we are saying X as a microfilter allows for free movement of the fetus as it grows and develops. Gaseous exchange between the mother and the fetus protects the fetus against desiccation, provides immunity for the fetus, meaning to say that the baby is protected from we are saying it increases the strength of the baby to fight against diseases, secretes a hormone thermoregulation, that is temperature regulation, thermoregulation, that is temperature regulation. So what is the question saying? Which of the following above which of the above functions is or applicable to the amniotic fluid? Write numbers in your answer book. We are saying the amniotic fluid, I had already mentioned some of the points there that the amniotic fluid, it allows free movement of the fetus as it grows and develops. So we can have numbers, this number, we can say 2. It allows this to take place. The amniotic fluid allows that to occur. It also allows protect the fetus against desiccation. We're saying drying out. It avoids drying out of the baby. which we are saying again is what? It's number 4, which is correct also. And then another question, another point is that it is also a shock absorber. We say that it protects physical and mechanical forces. Shock absorber, mechanical and physical forces. Physical forces which can impact on the mother. It is what? It is 7. And then we are saying thermoregulation, which is also 8. It also controls temperatures. It does not work, it does not provide immunity. Immunity does not come from what? It does not secrete any hormones. There is no hormones that are there. X is a microfilter, is not that. The microfilter, we talk about the placenta. So it is that 2, 4, it is 2, it is 4, it is 7, and 8. These are the functions of the what? Of the amniotic fluid. The diagram shows the human hormonal regulation of the female reproductive cycle. Hormonal regulation of the female reproductive cycle. So what is given here is that we have got these hormones that we have got the hormonal control or hormonal regulation. So pituitary or hypophysis hormone level, and then growth of follicles, ovarian hormone level, thickness of the of the uterus or of the uterine lining. So we have got days 0 to 28, and then we are seeing the growth of the follicle, which is also here. What we can see is that we can say this is where ovulation occurs, as we can see that the egg is coming out of the what? Of the ovaries. And then we can talk about the arrows that are there saying that we are saying these are also showing the difference, or we are saying hormonal control, and then the growth of the follicles. So let's see. We also look on the thickness of the what? Thickness of the uterine lining. It keeps on thickening. It started to thicken up from day 7, and then it keeps on increasing. It keeps on increasing, and then it is what? It is preparing a cushion. It is preparing a cushion there. So question saying, give the number and name of the hormone that stimulates the development of the follicle. The follicles are caused to develop. We are saying follicles is actually then give the number and name of the hormone that stimulates development of follicles. So the hormone which is responsible is follicle stimulating hormone. It is actually follicle stimulating hormone, which we can see that we have got. It is given here that when the follicle stimulating hormone it is high, we see follicles developing. So follicle stimulating hormone, follicle stimulating FSH, follicle stimulating hormone, which is 1 because it is high. So therefore, the more it is, the more follicles are growing. So we are saying it is 1, which is representing follicle stimulating hormone. Please take note that the question like this, you guys sometimes make mistakes. There are 2 questions there, the number and name. The question saying number and name. So please don't make mistakes of only giving one answer and then you lose 1 mark unnecessarily. And then maintains the endometrium between day 17, between days 7 and 14. Give the name and the hormone that maintains endometrium between days 7 and 14. So days 7 and 14, it is actually maintains the endometrium between days 7 and 14. It is actually because of the oestrogen hormone, which is oestrogen hormone. According to the graph, we must find that what is or which one is the oestrogen level. We are saying oestrogen, which is we are saying ovarian hormonal level. So this is and one key thing that I want you to take note of is that you need to know uh, as you can see, somebody can be confused to say, why did we say that follicle stimulating hormone is one? And then how did we choose that it is one in there? We are saying follicle stimulating hormone, it is produced from the pituitary gland. And then what happens is that it has to be more in order for it to allow these follicles to grow. Follicles development is caused because of FSH. So we are saying one, because that is where we have got the pituitary gland here. Our pituitary gland is given there. Also, we are looking at the one that is, according to this next question, to say that the, the hormone that is responsible for maintaining of the endometrium between day 7 and 14. We are saying this should be from the ovaries. So we are saying when it is high, it is there to when follicles are developing, and then we have also an increase in the secretion of what? Of estrogen. Estrogen secretion increases as the follicles develop. So it is also question, it is also that to say 4, it is from the ovaries, and then it is high from day 7 to 14. So it is what? It is 4, and then we say that it's what? Estrogen. It is 4, and then you know that it is estrogen. And then explain the function of the corpus luteum. Corpus luteum actually, corpus luteum, I have already answered something on it. Corpus luteum, what does it do? It is actually the inner membrane of the ovary that remains, which actually secretes progesterone. It secretes progesterone, which also enables the maintenance to help in maintenance of the endometrium. Endometrium is kept in position after fertilization by what? By progesterone hormone. And then question next, describe how the corpus luteum is formed. Corpus luteum, it is actually uh, it is formed, we're saying, after ovulation. So what happens is that it is formed after ovulation, which stimulates the conversion of the what? Of the ruptured graphical follicle, of the ruptured graphical follicle, and then uh, into the corpus luteum. You need to note that after ovulation, due to LH, luteinizing hormone, we are saying that which from the pituitary gland, it actually stimulates the conversion of the ruptured graphical stimulates the conversion of the ruptured graphical So now what you need to know is that the graphic it is the ruptured graphene follicle. It is actually converted into corpus luteum. Into corpus. It is actually after ovulation due to luteinizing hormone from the pituitary gland stimulates the conversion of ruptured graphene follicle into what? into corpus luteum. So it is ovulation that occurs, which actually enables the conversion of the graphene of the ruptured graphene follicle into corpus luteum. That is one key issue or one key point that we need to take note of. Give two visible reasons using the information from the diagrams and graphs above to explain why the woman is not pregnant. Why the woman is not pregnant. Why is the woman not pregnant? If you can see here, we are saying, let's look on the diagram, give reasons from the graph to explain why the woman is not pregnant. The pregnancy is not there because of what? The corpus luteum is shown there at day 14 after ovulation, which is forming there. And then we also see that number 5 represents the progesterone hormone. So what is happening is the progesterone hormone after pregnancy or after fertilization, if ever fertilization is occurred, progesterone hormone must keep on increasing and the corpus luteum, it must keep on growing. So what is happening is we are seeing a shrink or a decrease in size. The corpus luteum is decreasing in size. This corpus luteum is becoming smaller, is getting smaller here. So those are the reasons that we can see there. It is because one, the corpus luteum is getting smaller. There is evidence of degeneration of corpus luteum. Remember, we said corpus luteum cannot grow small because it has to keep on producing progesterone hormone. So the person is showing that she's not pregnant because there is no further secretion. Evidence of degeneration of the corpus luteum can also show that there is no pregnancy. That is showing the corpus luteum is not there, which is actually showing that. And then we can also see that there is also the drop or a decrease, which I also showed you here, that the decrease of progesterone hormone, this graph which is broken here, I'm talking about this graph, it must not turn down, it must keep on going up. It must keep on going up. That would represent that the woman is pregnant, but she's not pregnant because it is falling there from day 21. So we can see that progesterone level also dropping from day 21. It indicates that the woman is not pregnant. The woman is not pregnant also because we see the progesterone level. And then if we can look at this again, if we look at it, we say that the follicle stimulating hormone from day 28, it starts to increase again. We say that when a person is pregnant, follicle stimulating hormone is actually stopped from secretion. It is actually uh, cut off. So from day 28, we see an increase in what? In FSH. From day 28, we are seeing here, this graph shows that follicle stimulating hormone, it is starting to grow again on top of what? On top of the graph. So we also see an increase in the secretion an increase in secretion of follicle stimulating hormone from day from day 28. It is visible on the graph there. It is actually showing on the day 28. That shows that follicles are being manufactured and growing, which is not supposed to be the case, or it is not that case when a person is pregnant. There is a negative feedback that occurs whereby the progesterone levels or pregnancy will impede the development or the secretion of follicle stimulating hormone. So in this case, what you just need to see is that this is theoretical knowledge which you should just have. In any case, you should know that when a person is pregnant, we should see an increase in corpus luteum. The corpus luteum tissue should keep on growing. However, in this case, it is dying out. The progesterone level must increase, not decrease. We must see a stoppage in the secretion of follicle stimulating hormone. However, in this case, it is not. So these are uh, critical, uh, visible points that can actually direct us to correctly believe that there is no pregnancy. So we are given a male reproductive system again here, which we are given here again. I said that you should always be able to label roughly to say that what are these which you are given there, to say we have got the penis, we have got the corpus gland, we have got the prostate gland, we have got the testicles, we have got the bladder, we have got the vast difference or the sperm duct being shown by A there. So be able to know this. This you can label it at any given. Whenever given a chance, be able to answer this question. You should be able to answer them. So these are bonus marks anyway. So let's see the question. Let's see the questions that are there. The question saying identify the path labeled A. Identify the path labeled A and E. A, like I said, A, the path labeled A here, we can see this is the sperm duct, which we said is also called the vas difference, which we also said that it is also called vas difference. A is the sperm duct or the vas difference, and then it is the sperm duct, and then E, you can see that is the penis. E, we can see this is the penis there. We're saying that is the penis, which is quite visible there. We can see this the epididymis G. We have got we have got the testicles, and then we can continuously label them forever and ever. But we want to answer the question that we are given here. We want to answer the question that are in question for us to finish the question paper only. So let's go to the next question. Is saying explain the function of structure G. The function of structure G. Actually, G, what is the function of G? G, if you can look properly here, we are saying G, it actually identifies the scrotum. G, it actually identifies the scrotum. It actually identifies the scrotum. the epididymis is actually a coiled tube that runs around. We can say the epididymis can be this side. That can be the epididymis. But in this case, we are saying D is the scrotum. G is the scrotum. So the scrotum basically, as you can see here, the function of G, it actually protects the testicles. Protects testicles and actually ensuring that they are at right position away from the body. It ensures that the scrotum, the testicles, are at right position away from the body. The scrotum is an elastic also sac that adjusts itself up and down with response to the environmental or atmospheric temperatures to also regulate the the production of the protozoa. So the scrotum is actually there to protect them, the testicles, ensuring that they are at right position away from the body, which is optimum for the maturation of the sperm cells. And then the next question, name and describe the process which gametes are produced in part F. Gametes are produced in part F, the testicles. We're saying, remember this one, I said, if it is not specified, we say gametogenesis, but this one, it says it is in male. So in male, we say it is spermatogenesis. This is spermatogenesis, which we say that the formation of sperm cells, which is caused because of, we are saying because of the influence, because of the influence of, it is because of the influence of testosterone, what happens is that uh, because of the influence of testosterone diploid cells in seminiferous tubules. Diploid cells from seminiferous tubules of the testicles, the display cells diploid cells in seminiferous tubules, actually, which we are saying that they undergo meiosis. They undergo meiosis, which results resulting in formation of what? Of haploid 23 any cells containing half the number of chromosomes in the father, containing half the number of chromosomes that are in the father, containing half the number of chromosomes. That is why you see I've said that I've marked to say that is what is haploid, which is N or 23, and then diploid, which we said that this one you should also say 2N. In any case, diploid, you can as well say 2N. You can as well say 2N, which means that is diploid. So you need to take note of that and be able to understand it is the testosterone hormone that actually causes semperus tubules to allow meiosis to occur, resulting in the formation of haploid cells of the spermatozoa from the father, such that it can be what it can be able to be normal and fertilize the egg. I draw a diagram of a gamete produced in part F. The structure of a diagram of a gamete produced in F. So the structure of the gamete produced in F, we say that the question is asking for the structure of the sperm cell. They want the structure of what sperm cell is what we are supposed to draw here. The structure of the sperm cell is what we are supposed to draw. So this is also one other question, which is simple, which I want you to not to miss. We say that a sperm cell, it has got a head. Just that my drawings are not that good anyway, but I will try to draw something which can also be able to give you something like relatively correct diagram or a picture. The structure of the sperm cell, which we can say that you should be able to go through it. So now, we have got the acrosome. We said that the acrosome is there at the tip of the sperm cell to allow fertilization, dissolving the outer membrane of the egg, and then we have got this, as to say, is the what? Is the nucleus, and then we have got we have the medullary, or we have the mitochondria. We have the mitochondria, and then we have the tail. So in other words, I want you to understand again that the sperm cell can be divided into what? Into three pieces, which we can say this one is the head. This is the head, and then we can say that there is the neck, and then from here this is the middle piece. The middle piece also, and then we are saying, obviously, the last piece, which we are saying is the tail, which comes covering from here up to the end. This is the what? That is the tail. We say that is the tail. So basically, that is what you should know, to say that we have got a head, which is made up the acrosome, the nucleus. And then one other thing is that you need to know that this one contains the genetic information, contains the genes. We are saying contains paternal genes here. They are found in there. It actually contains the genes from the father, the nucleus. So again, the mitochondria, we said that it's for a refurbishment. It has got the energy for sperm movement. And then this one also provides locomotion or promotes locomotion because of the what? Because of the twist and turning of the tail. It also allows locomotion or the movement of the sperm cell. So one other thing is that the structure of the sperm cell comes saying, explain how is it structurally suited to perform its function. These are questions that you should be able to explain, that it has got the acrosome, which is at the tip of the sperm cell there, and then it has got the nucleus, it has got the mitochondria, it has got what. All those you should be able to explain and be able to say that when you are asked to explain the suitability of the sperm cell to perform its function, you should be able to say to explain those. So please, I wish you that you won't get confused. And during fertilization, the sperm cell cuts by the what? By the neck. During fertilization, it is only the head that enters the what? It is only the head of the sperm cell which enters the egg to allow fertilization. And then that is where now we are going to talk about the mitochondrial DNA coming from the mother and so forth. I'm sure that you won't be having any problems with that. And then the question is saying, draw a flow diagram showing the pathway of sperm cells during ejaculation. Draw a flow diagram to show a flow diagram. Draw a flow diagram. Here we are saying, please take note, the flow is just to show the sequence. It is a diagram that shows the sequence in terms of the pathway or in terms of the release of the sperm cell. Ejaculation, we are saying the release of semen of the sperm cell. That's what we call ejaculation. So now, what we need to know is that we can say the sperms are stored in the epididymis. Sperms are stored in the epididymis for them to mature. So we have got the epididymis, and then from the epididymis, the sperms are taken through the sperm duct. The sperm duct, sperms are taken, we see sperms are being taken to the what? To the urethra. Sperms are taken to the urethra. Therefore, we are saying this is how the sperms are released or are moved from the testicles into the vagina. We are saying that they are taken through the epididymis. They go through to the sperm duct or the vas deferens, and then to the urethra, which is the passage from in the sperm in the penis. A student studied the effect of various plant hormones on the growth of corn seedlings. Corn seedlings of the same height and species were used. The student took four groups of 10 plants each, 4 groups, and then all initial heights of the seedlings were 1 centimeter, meaning to say that they were measured. They were measured. Each group received the same amount of water. That is also maintenance of the experiment. She gave one group water only, while the other groups each received 1.00 micrograms of different hormones mixed with water. So one group was given water only. So after 15 days, she measured the height of each of the seedlings from each group, calculated the average, and recorded the information in the table as below here. So various plant hormones and mixed in water, height of the seedlings in centimeters. So we have got water only is 21 centimeters. We have got abscisic acid plus water, 19 centimeters. We have got oxins plus water, it is 22 centimeters. We have got gibberellins plus water is 48 centimeters. Also, please refer to the channel Metric Hub for plant growth hormones. They are well explicitly explained. Plant growth hormones, where are they produced, their functions, how do they work in terms of the breaking of dormancy and promotion of dormancy. All those are given on the channel. Please go and verify on that and get empowered right now before our exams on Friday. So the question is saying, which plant hormone produced the tallest plants? Here we are not going to calculate or work out anything. We just look on the centimeters that we are given here and we get the what? The highest. The highest is the one which has got the tallest plants. We get them from the highest what? From the highest. So this is just given that it is what? Gibberellins, which is which plant growth hormone produced the highest. So it is gibberellins is given there. Gibberellins, they have got 48 centimeters, which is the tallest, which is given there. And then identify 2 ways that the student ensure the validity of the investigation. The validity of the investigation. I was marking these as I was giving you this question. I was reading through. The learner or the student used the same. The learner used, he actually used the student took 4 groups of 10 plants. He also, the student studied the effects, and then same height and same species were used. So one, the student used same species of plant were used. Same species of plant were used. We can see that it is given in the essay there. He used also same number in each sample, where he said student took 4 groups of 10 plants each. He took 4 groups of 10 plants each. Used, so we are saying the student same number of seedlings used in each sample. Same number of seedlings used in each sample. We also have the learner also she also same height of plants were used, same height, which we said all initial height of the seedlings were 1 centimeter. So same height of plants were used. Same height of seedlings was used. We can also have the same, he's saying after, I see that the learner said after 15 days, she measured the height of each seedlings from each group. So 15 days were given to each group. 15 days were given to each group. So we are saying same amount of, same period of time was given to the experiment. Same amount of time was given, which is 15 days each. It is given in our context there that they were given 15 days each. After 15 days, he also did that. And then I see again, she gave one group water only, while the other groups each received 1.00 milligrams of different hormone mixed with the water. So she also used we also talk about same amount of hormone mixture in water was used, same amount of mixture, same amount of hormone mixture, same amount of hormone mixture was used, which is given there to say that 1.00 was used. All these are also points with regards to the validity of the investigation. So sometimes again, we can also even talk about when these questions come, you might also need to know that questions like these requires you to know that when we are doing an experiment, we need to ensure that the results are very dependable, sorry. They are valid. So in terms of that, we consider all these points that we have mastered and listed here, give two functions of abscisic acid, give two functions of abscisic acid. So number 2.33, abscisic acid, it actually controls loss of water vapor. It controls loss of, let me just say it controls transpiration in plants. It controls transpiration. It controls transpiration in plants by controlling the opening and closing of the stomatas. We are saying opening and closing of the stomata. The opening and closing of the stomata is actually affected or controlled by what? By 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 this abscisic acid. It also stimulates the falling of leaves. It stimulates the falling of leaves or of fruit. Falling of leaves. It stimulates falling of leaves, or we can say fruits in some plants. We can also talk about the abscisic acid. It also promotes flowering in some plants. Promote flowering in some plants. Some plants do flowers because of abscisic acid. And then it also promotes dormants, inhibiting germination of seeds. Inhibits germination of seeds in some seasons. In order to avoid seeds to germinate prematurely or to germinate in the wrong times, abscisic acid, it actually inhibits that. That is called promoting. It is called promoting seed dormants. It induces plants to. It enables plants to survive erratic conditions. Times when things are not good, plants are able to survive because of the abscisic acid. These are quite a lot of points. Please just go also on the channel. You can find as many other points as you want. And then it is draw a bar graph to represent the data on the table above. The diagram that can be drawn to represent the data on the table above. So we are going to draw a graph that is going to show we are talking effect of plant. We want to look on effect of various plant hormones on growth of corn seedlings. So we say that the graph must have a title. We can talk about effects. Title. It is a bar graph showing effects of various plant hormones, effects of various plant hormones on the growth of corn seedlings, on corn seedlings growth. So this is the first thing that you need to take note of. The bar graph, it must have a title. So bar graph, that must have a title. So we have to look on our dependent and independent variable. We say that the dependent variable is on the vertical axis and independent on the horizontal. So what is going to be on our vertical? So we see this must be on the vertical axis. It must be on the vertical axis so that it can control the shape of the bar graphs. And then obviously we have got this on horizontal axis. Horizontal axis, we have got what? Various plant growth hormones in the horizontal axis. So we also look on the set of data, the highest and the least. The highest is 48. We are saying the highest is 48 and the lowest is 19. So we need to look on that and draw a paragraph with a scale that can be able to make us to see what is happening. So now what is happening is we have that title there, and then we can say on our vertical axis, and then we have got our horizontal axis again. So here I said that we have got we said on the horizontal, on the vertical axis, we said we have got height of seedlings. On this one we have got height of seedlings is given on the horizontal, on the vertical axis, and then we said on the horizontal we have got plant hormones mixed with water. We have got plant hormones mixed with water. Plant hormones mixed with water. So that is on our horizontal and on the vertical like that. So we need to look on this. Remember that this is a bar graph. So we just have to look on each of the we said the highest is 9, the lowest is 19, and the highest is what is 48. So we can choose a proper interval that we can use, which we can use there, which we can say, if I can say you have to find a proper interval that we can use for this graph. So if we say 10, 20, 30, 40, 50, 60, 10, 20, 30, 40, 50, 60. So now what is important is that this interval class is very important because it enables your graph not to be too big or not to be too short again. And for you, remember that you are drawing this graph with a ruler. You don't just draw with a free hand. Now I can use a free hand because right now I'm just sketching. So you need to use a ruler. So now we are saying the first one is water only, which is 21. Water only, it is 21. We go on water only, 21 centimeters. 21 centimeters. So don't forget again to indicate the what? Don't forget to indicate the units here to say these are in what? In centimeters also. Don't forget to indicate that. So we are saying 21, it is water only. I can say 21, which we can say it is what? It is water only, which is 21. I can just draw it like that. And don't forget to indicate that, don't forget to indicate the measurement is 21. And the next one is what? Is 19 abscisic acid plus water, 19 abscisic acid and water. I can come here say that abscisic is 19, it's abscisic plus what? Plus water. Abscisic acid plus water. When you write in full, now I'm just writing in short to say abscisic acid plus water is what we have there. And then we have for the next graph, the next bar, it is 2248. 22 is oxygen and water. 22 is oxygen and water. We have 22, which is oxygen and water. We are saying it's oxygen and water. We are given that this is oxygen and water here. We are drawing your graph, oxygen and water. Oxygen and water is the same way we did there. And then lastly, we have got 48, which is gibberellins. 48, which is gibberellins. We have to go. That is what 48, which we are saying is gibberellins plus what? Plus water. So this, we can just also just mark them gibberellins, abscisic and water only. So these are our graphs that you can see here. So you can see this is our graphs. So, and again, I said don't forget to indicate the measurements. To say this is 19, this is 29, this is what. It's very important that you mark these in order for the examiner not to think otherwise because of your graphs. I cannot be, you are only indicating that the measurements that you are putting here, the readings are very important. These readings are very important. Readings to mark height of bars. Height of bars is very important. Readings to mark height of bars is very important. You need to put on each bar the readings that are given there. So when you do this, you have already scored all your marks. Title is there, labeling of the axis, the bar graph is also marked. So therefore, there is also a scale of 1020. That is a scale that we are talking about. So it makes your graph to be what? And your marks to be quite explicit. You won't lose any marks on that. We are given here is the structure of the human eye, and then name the part. We are given the structure of the human eye, which is asked to say give the name of the part that maintains the shape of the eye together with the sclera. The name of the part that maintains the shape of the eye together with the sclera. We are saying this question, you would always find out that the shape of the that maintains the shape of the eye together, the shape of the eye together with the sclera. So you should talk about this part. You should talk about this part here. The shape of the eye is maintained because of this part. This maintains the shape of the eye. It maintains the shape of the eye here. Shape of the eye is maintained here, which we are saying it is the what? It is the vitreous humor. Vitreous humor. The vitreous humor maintains the shape of the eye. The vitreous humor is that liquid that is inside the eye. It is the vitreous humor, carries impulses to the brain. I give the name of the part that carries impulses to the brain. It is the optic nerve. The optic nerve carries the optic nerve. It is the optic nerve that carries impulses to the brain. It is the optic nerve, which we say that in that position we find out that that is where impulses are carried. And then explain one feature shared by part B and D that make them suited to function for clear vision. Part B and D, part B and D, part B, part B and part D. We are saying part B and D. The question is saying explain one feature that is that shared by part B and D that makes them suited to function for clearer vision. So one, both they allow reflection. As you can see, we said that part B and D, we are saying we are saying the cornea and so forth. What happens is that they both have got a convex shape to allow reflection. Both allows reflection. Both allows reflection of light uh, by their convex shape. They both allows the reflection of light because of their convex shape. Also, we also have they are both transparent. They are both transparent. Allow to in order for the light to reach retina. We said that in order for us to see, the light must reach the retina. So the image must fall on the retina. So what we are saying is that they are both transparent, that allows light to reach the retina, and also they both have a convex shape that allows refraction or bending of light. They both allows light to bend towards the yellow spot or the retina for clearer vision. And then discuss how part G and H work together for vision to occur. Part G and H. Part G and H. What happens is that we are supposed to look. We are saying G and H. How do they work together for vision to occur? Part G and H. We are talking about this to say how do they work together to allow clearer vision. So what we need to understand is that we are talking about to say what happens in order for this to have for us to have clearer vision. So we talk about the issue of the photoreceptors that are found in G. There are photoreceptors here. Photoreceptors are present in G. And then what happens is that what happens with the photoreceptors here, they actually they are able to convert light to impulses, and then H is actually transferring. It is the optic nerve which moves the impulses to the brain. So now what we are saying is that we need to understand that G, it is actually the retina or the yellow spot. You first of all need to also find out that is the retina, or we can say that it is the what? It is the yellow spot. So all that you need to take note of. So what we are saying is part G, we are saying the retina, yellow spot is photoreceptors. It has photoreceptors that convert light into impulses to convert light into impulses, convert light into impulses, and then the impulses are transferred to they are transferred to optic nerve, which we said H. They are transferred to optic nerve, which we said H there, so that they can be carried to the brain. They can be carried to the brain. So what we are saying is that questions such as those, they are very important. They are very important for us to understand, which we say that we need to know that we say that it is the what? The cerebrum for interpretation. They are carried to the cerebrum, which is the what? The brain for interpretation. Therefore, we can talk about the functioning of this. We can talk about those as a function. And then tabulate two structural differences between a healthy eye when looking at an object closer than 6 meters away, and when looking at a further object at an object further than 6 meters away. So class, what we are asked about here is to explain far and near vision. That is what we are talking about, whereby you need to look at what? It accommodation. Accommodation ability of the eye to look at near and far vision. That is accommodation. So now, what we need to look at now is that we need to differentiate between near and far vision now. So object closer than 6 meters and object further than 6 meters. That's what we need to do. Say object object closer than 6 meters, which we are saying this is near vision. And then we also go to say object far than 6 meters, which is far vision. That is what the question is asking. When they are saying tabulate, it means the moment you do this table, you have already got yourself the mark for being able to tabulate. You have got a mark already to be able to tabulate. So one we are saying a lens, they become more convex on closer vision. Lens becomes more convex. However, lens become convex. Becomes less convex. There is more, there is less. And then two, we must say the ciliary muscles contracting. The ciliary muscle contracting, and then at this side, the ciliary muscle it relaxes. The ciliary muscle relaxes on the other side when it is a far vision. You see, what is very important here is for you to understand the reason for this, to say that when an object is too far, more light must be able to enter the eye, and when it is too near, we also need to reduce the amount of light that is entering the eye. So now, those are the basic understandings that you need to look at. And then suspensory ligaments, they become, the suspensory ligaments become slackened. Suspensory ligaments become, they become slackened, meaning to say that they are not pulled. They become slackened, meaning to say that they become slacky. And then what you need to understand is that in that case, the suspensory ligaments, they become tight in the other side on a far vision. Suspensory ligaments become they become tightened. They become tightened. That is what happens there. So at the end, we say the lens, therefore, finally, in other ways, this must be the last point. The first point must be the last point of the lens, therefore becomes less convex, and the lens becomes more convex. That must be the last point if you can follow it properly here in terms of the sequence of explanation that must be explained, that is expected of you. So now, certain times the question wants you to say you need to talk about only, you might be asked to talk only about this, about short-sighted, about near vision. You might be asked about near vision alone, or you might be asked to talk about far vision. When they say more than 6 meters, it means you are going to talk about this one. When they say less than 6 meters, it means we are going to talk about this one. And then sometimes there is a diagram that can come. So when we go to this one, let's also look at diagrammatic diagrams explaining or illustrating this, or illustrating this explanations. It's very important for you to know the diagrams. When diagrams are drawn, you should see that when we are talking about near vision, you shall see that when we are talking about near vision, we shall find out that we talk about suspensory ligaments. They are slackened, and then we are saying the lens is more convex. That is it. And then when we are talking about the opposite, which is far vision, you shall see that we'll talk about something which is showing almost a pulled diagram like that. This you can see they are what they are tight, and then the lens is what the lens is less convex. This is the lens, and then you can see that it's less convex. And then we have got tight suspensory ligaments. That is there. So what I'm saying is you shall see that these diagrams also are very important for you to be able to understand this, to say which diagram explain, identify the process shown there. So this is near vision. And then obviously the other one is what is far, as far as you might need to understand or to be able to expect any possible ways of questioning that might arise there. We are given an investigation was conducted to observe the time taken for blood alcohol levels to return to normal after consumption of various volumes of alcohol. And then the investigator invited 4 people to participate in the investigation. Invited 4 people to participate in the investigation. Each participant was asked to read the following governmental warning label about consumption of alcohol. So we are given that according to the Surgeon General, women should not drink alcohol beverages during pregnancy because of the risk of birth defects. It actually affects children. And then consumption of alcoholic beverages impairs your ability to drive a car or operate machinery and may cause health problems. They are the effects of alcohol that are given here. Each participant was given a specific volume of alcohol to drink. So participants 1 to 4, each one was given 30, 60, 120, 180, which is showing an increase. It's 30, it's 60, 60, 120, 122. This one is 60 again. So the blood glucose concentration was measured before the participants drank the alcohol, and every 30 minutes after until the blood glucose blood alcohol concentration returned to 0. The graph shows the results of the investigation. We are given the results of the investigation here. The graph shows the results of the investigation. So now, what is happening is we are given that alcohol concentration in blood glucose in percentages that is given on the vertical axis here. And then we are given time here, range of common legal limits on motor vehicle operation, which is given there. So let's see the question. What is the question saying? It's saying how many hours does it take for a person to be legally able to drive after consuming 180 mils of alcohol? If a person drinks 180 mils of alcohol, how many hours does it take for the person to be legally able to drive? So we go to 180. We are saying range of common legal limits is this one. So 180 is when a person drinks 180. We are seeing alcohol concentration, it is increasing there. And then we are saying he drinks from here. And then this going back to normal is somewhere here, which we can see it can be, which we can see that it can be 2.3 to 2.4. 2.3 to 2.4 hours. Here, we are looking at this level where the person is able to go back to normal. So the normal time is here. We are saying at the normal time, it is here, which is, according to, we read the horizontal time there. So which is 2 hours, 40 minutes. Two hours, 40 minutes. We can see that from the group, from the graph there. And then from the graph, identify the dependent variable. I said these questions are very easy and you must not lose any marks. Identify the dependent variable from the graph. Dependent is on the vertical axis. That's what I always tell you. The dependent variable is always on the vertical axis. So, I mean, the question is saying identify the dependent variable from this one. The dependent variable, we are saying that it must be found on the alcohol concentration. We are saying it is alcohol concentration, which is on the vertical axis. I said dependent is found on the vertical axis class. This is the dependent variable. It is dependent. Dependent affects the shape of the graph. It is the one which is dependent on time. So we are saying the dependent variable is alcohol concentration. It's actually blood alcohol concentration. Blood alcohol concentration is actually the dependent variable. And then suggest 2 reasons why the investigator would ask the participants not to consume alcohol for 48 hours before the investigation. This is actually to ensure that he actually wants to increase he actually wants to increase or to ensure the validity of the investigation, that they must not drink alcohol 48 hours before the investigation to ensure that their body is clean. So, one, to ensure validity of the investigation. To ensure validity of the investigation. Also, he wants to ensure that ensure all participants have same amount of alcohol, same amount of alcohol, to ensure that the participants have same amount of alcohol. Actually, that ensure they've got same amount of alcohol or zero alcohol concentration, same amount of alcohol concentration. He is trying to ensure that the concentration of alcohol in the blood of these guys or in the blood of these people are the same. In other words, we can say that he wants the alcohol concentration to return to zero. He's giving enough time for alcohol concentration to go back to normal, to zero. Allow the blood concentration of alcohol to go back to zero. It can be also a possible reason why he's giving 48 hours to allow alcohol concentration to go back to zero. That can also be the reason why he's allowing 48 hours alcohol concentration to go back to zero. For this investigation, describe 2 safety precautions. I once also said that questions such as this paper that we are doing is quite exemplary. That questions such as this would come, safety precautions and then planning steps. So, Safety precautions in terms of this: one, ensure that that all participants started the investigation with no alcohol in their blood. Ensuring zero alcohol in all participants' blood. In all participants. All participants must have zero alcohol in blood. Is very important, that one. And then ensure that the participants are not allergic and they have no adverse effects to alcohol. Ensure all participants are not allergic to alcohol. Remember, when we do an investigation, it is always very important and profound that when you do investigations, your experiments must not interfere with the living organisms that we are using. So in this case, there shouldn't be anyone who can suffocate or die because of taking alcohol. That is very important. So therefore, he has to ensure that all participants are not allergic to what? To alcohol. And then participants must not operate machines or drive. It is given in context that participants must not range of alcohol limit, concentration returns to zero, and then we are told that the participants are not supposed to, also we are saying they must not drive after taking alcohol. So these are all precautionary measures that we are put in place. Participants must not operate machines after alcohol or after taking alcohol to avoid. We are saying these are safety precautions. They must not drive as well. And then planning steps. Planning steps. The planning steps that are there. The planning steps that are there. We are saying when you are given an experiment, you are always supposed to know what equipment you are going to use. So you are going to decide on data. We say you can decide on data recording procedures. Deciding on data recording methods. Data recording methods. You can also decide on time of the investigation. Deciding time of the investigation. People must know when are we going to do the investigation. The time of the investigation must be decided. And then getting permission from the participants. Getting permission from participants. People that they are going to use in the investigation must consent that. Getting permission from participants is very important. Getting permission from participants is very important. We can also talk about identifying the age and the number of participants to use. Identifying the number and age of participants to use. These are all planning steps that can also be put into account when looking at planning steps. And then based on the table above, are the investigation results reliable? We are saying based on the results of on the table, the results based on the results on the investigation, we're saying that the results are based on the table above, based on the investigation, the results can we say that they are reliable, these results? Can they be reliable? We want to look on the results on the table. We see that number participants, we have got 1, number participant 2, participant 3, participant 4. So what is happening is it cannot be reliable because the sample size is very small and the investigation was never repeated. It's not reliable. The results, they are unreliable because the sample size was too small. Only 1 participant per each was too small, and investigation was never investigations were not repeated. So we can say that the results are unreliable because the sample size was too small. Remember, we said that in order for us to get reliable results, we need to have a big sample size. At least about 10 people are given the alcohol, and then we are able to see the changes and the response to that. Give a reason for your answer. We already mentioned some of the points already. We can also say that the higher the volume of alcohol consumed, the longer it takes to reduce blood alcohol concentration. The higher the volume, the higher the volume of alcohol consumed, the longer it takes to reduce blood alcohol concentration. The longer it takes to reduce blood alcohol concentration. That is one other reason again. And then we can say a suitable conclusion for this one. A suitable conclusion here is again what we have already mentioned, that a suitable conclusion is what we have already mentioned in this respect, that the higher the alcohol concentration. If you can check on the tab on the table here, we are saying volume 40 alcohol. We are saying if you look on this, the more the volume, the more the alcohol drunk, the more the time it takes for the person to get back to normal. We are saying 60 mils, it just goes back. 120 mils is just, but 100, we are saying 180 mils. The more the alcohol are drunk, the more time it takes to go back to normal. The more time it takes to go back, the more alcohol, the more time it takes to go back to normal. I've shown that through those graphs. And then AirPods. AirPods are wireless listening devices that fit into the ear and direct sound, allowing users to listen to music from their phones privately without disturbing others. And then diagram one, we have got diagram one, we have got the earpod. As you can see, I think you are using these earpods. They are put in order for somebody to listen to sound or music without disturbing people around. So smartphones, yes, a feature that can monitor the volume of music being played through the app, the AirPod, and give warning notifications if the volume exceeds 80 decibels. So Fred and Tebuho are listening to the same music. Diagram 2 and 3 below shows their phones display. So we are given here diagram 2 for a dispose the AirPods 80% battery, 98 sound level, 92 loud is danger. It shows danger here. It shows danger. And then we are seeing here table AirPods 100%. I mean AirPods, we are saying 100% battery, sound level 34 decibels, and then it is okay. So now the question is saying name the part of the ear that vibrates when the sound name the part of the ear that vibrates due to the direct effect of sound waves. I'm sure that you know that it is the tympanic membrane. Tympanic membrane actually is responsible for that. The tympanic membrane is actually responsible for the vibration, which we said that sometimes it can be affected because of the hardening, which can be caused by mucus, which we can found in the ear there. And then name the part of the ear that contains the receptor cells which detect the sound stimulus. The sound stimulus is detected by organs of Koch. Organ of Koch. And then equalizes pressure in the middle ear. I'm sure I remember this one. It connects the ear to the throat. It is the eustachian tube. It is the eustachian tube which is connected from the throat to the what? To the ear. It helps to equalize pressure there. Channels sound waves to the middle ear. It channels sound waves to the middle ear. It is the auditory canal. Is the what? The auditory canal. It is responsible for that. And then explain 2 ways in which AirPods could prevent a person from hearing other than environmental noise. So now, what happens is that one, we are saying other than prevent a person from hearing other than environmental sound. The AirPods block the auditory canal and prevent the sound waves from the environment from reaching the tympanic membrane. Sound waves are blocked from reaching the tympanic membrane, which we said that is responsible for vibration reaching tympanic membrane. That is one other way. And then we can also sound waves from the airport is too loud. Sound waves from the airport is too loud. It's actually too loud, and the environmental noise is too quiet. It's too quiet. So that is one other way that we should look at that. And then use diagram 1 and 2 and answer the following questions. Let's see diagram 1 and 2. Who is at more risk for damage of their sound receptors? If you look on this, who is vulnerable to damage of the ear receptors? We are saying who is responsible for damaging of ear receptors. We are saying Freddy and Tebo are listening to the same music. Diagram 1 and 2 below shows their earpod. So now, Freddy, we can say this is Freddy and this is Tebuho. That is Freddy and Tebuho. So what we are saying is that this one, which is actually saying too loud and 92 decibel, so is the one that is at what? At risk. So who is at risk? It is what? It is Freddy. It is Freddy there. It is Freddy. And then why do we say Freddy? We are saying, actually, if you can see, I said that Freddy because we said the sound, it is even given that too loud. And this one, sound level, it is called okay, therefore it cannot cause. So Freddy's sound is what? It's more than safe. Freddy sound level is more than normal, is more than normal, which we are saying the volume is given there that it is 92. It is given that it is 92 dB. And then we are given the diagram below shows the male body with the position of various glands. Position of various glands are given here. And then the question is saying, we are given this. I said that this question, it is very common for it to come. It is very common for it to come because you should be able to identify glands and the hormone that they produce. So name the hormone secreted by the adrenal gland. The hormone produced by adrenal gland, it produces what? Adrenal gland, it produces, it actually secretes adrenaline. Adrenaline, and also it secretes aldosterone. And also aldosterone. These are hormones of the what? of the adrenal gland. And then name the hormone secreted by gland G. G is the testicles. It secretes what? Testosterone. Testosterone hormone is the one that is secreted from D there. It is the testicles here. It is the testicles. Therefore, they secret what? Testosterone hormone. They secret testosterone hormone there. And then name and describe the role of the gland. Name and describe the role of gland A when the hormone secreted by B increases above normal. Name, it is saying name and describe the role of gland A when the hormone secreted by B increases above the normal level. So gland A, name and describe the role of gland A. Gland A, it is actually, we can say this is the hypophysis, is the pituitary gland. It is the pituitary gland. So it's saying name and describe the role of gland A that when the hormone secreted by gland B increases above normal, which is when gland B that is we are saying the thyroid the thyroxine hormone. Thyroxine is this is the thyroid gland. So what are we seeing here? We see thyroxine hormone is secreted there. So now we are just supposed to talk about this one. We have to talk about the relationship between the pituitary gland and thyroid stimulating hormone and thyroxine. So it is pituitary gland, the pituitary gland. So the raw pituitary gland secretes a thyroid stimulating hormone. It secretes thyroid stimulating hormone into the blood. It secretes thyroid stimulating hormone into the blood, and then low thyroid stimulating hormone, the low thyroid stimulating hormone levels, the low thyroid stimulating hormone levels actually stimulate the thyroid gland to secrete thyroxine. The low thyroid stimulating hormone levels actually stimulates the thyroid gland to secrete less, to secrete, to start secreting less of thyroxine hormone. Less of thyroxine hormone, and then resulting in its volumes. Resulting in its volume decreasing, and then therefore it goes back to normal. So the question is basically asking about the balancing It wants you to understand the balancing of what? The balancing of thyroid stimulating, the balancing of thyroxine level in the body or in the blood. How is thyroxine level balanced? So if less thyroid stimulating hormone is secreted, it means it would also, if we have got less of thyroid stimulating hormone, it means what is happening is that the thyroid gland is also it is also stimulated to secrete less of thyroxine hormone. Thyroxine hormone is caused to be secreted because of thyroid stimulating hormone. So if it is less, it means also less of thyroid. If less of thyroid stimulating hormone is secreted, it also means less of thyroxine hormone is also produced, which results in its decrease and going back to normal. Uh, let's just answer the last one. This is going to be our last question. Fazin mass 70 kgs, non-diabetic. Aubrey mass 70 kg, diabetic. And then period of exercise, normal range. And we are given time and blood sugar levels that are given there. So the question is saying, name the hormone Aubrey. Name the hormone Aubrey received at X on the graph. Name the hormone that Aubrey received at X on the graph. Aubrey is this one, and then he received a certain hormone at X, which we are saying the hormone, it actually caused the normal, the sugar level to go to normal. So the hormone which controls sugar level is what is insulin. This is what insulin. Insulin, because it's the one that reduces or it converts. We are saying this converts excess glucose or controls glucose levels in the blood. As you can see that the blood glucose level is therefore decreasing, and therefore this is what? This is insulin. That is insulin. And then provide a suitable reason for your answer. Provide okay. Why do we say that is insulin? We already talked about that converts excess, because when Aubrey was given the insulin, the level of blood sugar, the blood sugar level started to decrease as well. So we are saying blood sugar level starts to decrease after administration of the hormone. Blood sugar level of Aubrey started to decrease, which also shows that it is what the blood sugar levels for Aubrey started to decrease. Therefore, we can say that the blood sugar levels started to decrease. Therefore, we can say it is insulin that converts glucose into what? Into or which regulates the level of glucose level. And then the question is saying, define homeostasis. Homeostasis is actually the ability of the body to maintain a more or less constant internal environment, regardless of changes in the internal and external environment. Homeostasis, ability of the body to maintain a more maintain to maintain a more to maintain a more or less constant environment regardless of its internal or external conditions. So what happens is that homeostasis, the body strives to maintain a more or less constant environment without being affected by the external factors or external conditions. So when temperature goes up, the body has got the capacity to return it to normal on its own. We say that that is homeostasis. So that's what you call homeostasis. So please understand that this is the effect of strenuous exercise on athletes' body temperature followed by ice bath. So we are given that the question is saying, the person is saying which part of the brain responds to changes in body temperature? Which part of the brain responds to changes in body temperature? The body temperature is controlled by hypothalamus. Hypothalamus is responsible for responding to changes in body temperature. And then the question is saying, describe the changes in athlete's body between 20 minutes as illustrated there. This question, class, it just wants you to describe the changes in athlete's body temperature between 20 and 40 minutes. This one, between athlete's body temperature, between the body, we are saying athlete's body temperature between 20 from here to 40 minutes. From here to 40 minutes is what is being asked of you to do. So here, what is happening is that we are seeing a decrease in body temperature, a decrease in body temperature is seen. As you can see here, the graph is going down, and then what happens is that the body temperature, we are seeing strenuous activity. The person has done activities too much from somewhere like 5 to 10 minutes. So because the blood vessel of the skin, it is because we are saying decreases, or the temperature decreases, but decrease in body temperature, and then blood vessels of the skin actually they vasodilate, the wide open, remember, to allow blood to flow at the surface of the skin. So as you can see, the temperature decreases, and then more blood flows at skin surface, and then resulting in a loss of heat by evaporation. the person has been sweating, and then heat is therefore lost by evaporation. And then we can also talk about more blood flows to the sweat glands, and then evaporation or evaporative cooling is the one that is causing the temperature to keep on dropping. The evaporative cooling results in loss of in terms of results in what, in that temperature keep on dropping, as we can see there. And then the last question is saying explain the cause of the last question is saying explain the cause of increase in the body temperature during strenuous. Explain cause of increase in body temperature during strenuous activity. During strenuous activity, the temperature increases. During strenuous activity, the temperature here is given. The temperature goes up. So what is the cause of that? It is because of there is an increase in cellular respiration, which produces more heat energy. I think you remember that respiration because of an increase in level of cellular respiration, which results in more heat energy being released. I'm sure I remember that we said that this can be explained as respiration. Respiration is equals to glucose plus oxygen, which is equals to carbon dioxide plus water plus energy, which we say the energy is heat, which is causing temperature to go up. This is what is happening. So I wish you all the best in these exams that we are to write now. I think what we have done today, like I said, is the last paper, is the last paper, is the last paper. So please follow it to the end like we did now. We have tried to work it and take more time on it so that we exhaust the other possible angles of questioning that might arise in our examination. So I wish you all the best. Let's look forward for paper 2. I'm going to bring to you a very, very standard and typical uh, question paper again that we are going to work on. Paper 2 is loading now. Check for it tomorrow. It's coming. Paper 2, prepare enough. Look for it. It's going to be available for you to also for you to make reference on. Have a beautiful time and all the best. Let's keep connected even in other upcoming subjects. Geography is coming. We are going to work it together. Economics, we are going to work together. All of them. Let's work together until we are done. Future leaders are born in Metric Hub. Let's do it now. Through a click, you can change your life. Keep connected until we meet again. Bye.